Thank you for joining us today for our webinar on addressing microbial concerns in pharmaceutical water systems. I'm Rich Jarrett, and I am Evoqua's market lead for the pharmaceutical segment. I am joined by Gary Zacalante, who will be addressing the many questions and concerns we've heard from pharmaceutical companies recently. Gary has been intimately involved with pharma water systems for over 40 years and has been active in the development of the many standards and guides that are used by the industry today. I have been involved with the pharmaceutical industry for over 14 years, providing products and solutions that deliver improved quality and manufacturing efficiencies. I believe you will find today's topic of extreme interest. We will review the rules and regulations around microbial control and what you need to consider when designing and operating your water system. In addition, you can also get access to our previous webinars on sanitization and membrane Wi-Fi production by accessing the Evoqua website or contacting me directly. We will have your phones on mute while we go through the presentation. Please feel free to submit your questions by clicking on the question button as we go through the presentation, and we will answer as many as time permits after the presentation is over. We will follow up with any questions we cannot get to. You can also reach out to me directly, and my contact information is included at the end of the presentation. Let me now turn the presentation over to Gary.
Thank you, Rich. I'd like to thank everyone who's on for being with us. We certainly hope to give you some valuable information that you can use. So what we're going to cover today, we're going to talk about how you determine microbial limits for pharmaceutical water, the impact of the sanitization method or methods chosen, when a microbial limit or limits are selected, what's the impact of that on system design, what are the microbial control factors that are going to be implemented to have a successful system, and then a more detailed level look at system design. And an hour is a short time for this amount of material. We could fill 8 hours with it. So we're going to go at a very fast pace, but I think we'll be able to give you a good overview. So first, we're going to talk about regulatory requirements and risk assessments. So how do you determine the microbial requirements for your water system? You need to know the product types produced and the use of those. You need to, or your team has to be able to do a patient risk analysis from that product in use. What's the point of manufacture? Where are the products shipping? What is the water used for? It could be for active ingredient production, rinsing, it could be in a dosage form. Even if it doesn't remain in the product, such as in a tablet, the water still falls under good manufacturing practice. In addition, you're going to have to know the pharmacopoeia limits for the water. So that's typically purified water or water for injection. There may be additional regulatory documents to consider. Some of those could be legally binding. Some may be non-binding, but in saying non-binding, if it is a regulatory guide that could be applicable, you're going to want to know what's in it and understand should you follow it, or if you don't follow it, how you could explain why you don't need to, or you're doing something better. Those are external requirements. The internal quality requirements within a company are frequently much stricter than the external. And I might add on the external, those are maximum action levels. So you need to understand your product and manufacturing to know if you need to be tighter than you're allowed by regulatory bodies. So internal requirements to set those, it requires knowledge of the manufacturing process. It's an understanding of the company risk aversion, and that is going to vary dramatically among companies and products. Generally, to get a more robust system, it's going to cost more money to do it. Ideally, if you spend more, you end up with a more robust system. The worst would be to spend money needlessly and still not have the robustness that you desire. Thanks to A.M. Condell for this slide, but it's an overview of products. And so parenterals, as an example, aerosols, nasal sprays, those are high up from a patient risk standpoint, where liquid-filled capsules, oral tablets, etc., are at the lower risk. So this may help you to start with an understanding of risk. Now we're looking at biofilm formation, and this is intended to give you respect for the adaptability of bacteria. That even with polished materials, it's possible that irreversible attachment could happen within minutes, or worst case, hours. That then growth and division of bacteria, hours to days. The exopolymer substance formation, hours to days. And so now you're beginning to get a tenacious biofilm. And that's what we're going to work to do, to eliminate it in some cases and to minimize it in other cases. Getting into pharmacopoeia requirements, the current USP Pharmacopoeia, edition 43, in the purified water and water for injection monographs, that's the legally binding section for water. Interestingly, the microbial limits are not stated there. So you might say, well, there's no legal requirement. In the general information chapter 1231, that is where purified water is shown at less than 100 colony forming units per milliliter. Water for injection is less than 10 CFU per 100 mil. But everyone accepts these as actual limits. In the 1993 FDA Guide on Inspections of High Purity Water Systems, FDA stated that these levels, which remain current from that time, they actually take them to be in the monograph or not as actual limitations. If we look in the Pharmacopoeia, or European Pharmacopoeia, there we have the same values, 100 CFU per mil for PW, 10 per 100 for water for injection. Those are in the monograph. And looking at, there are over 140 Pharmacopoeia worldwide now and ever growing. And in most of those, the microbial limits are in the monographs, making them clearly legally binding. And fortunately, there's excellent harmonization worldwide on these levels for purified water and water for injection. The next topic, objectionable organisms. This may apply to you and your company or not. So as an example, in 21 CFR 211-113 for non-sterile drug batches, you have to assess the drug administration, the product, the patient risk. And do you need to prevent objectionable organisms, organisms that could cause infection, that would present patient risk? So this is not telling you that you do have that issue. It's telling you you need to assess it. You need to see if you have the issue and then how you would demonstrate that it's addressed. In that general information chapter in USP 1231, the same topic is discussed. And generally, it's gram-negative bacteria that are typically the bacteria of concern. There are two other USP sections, 60 and 62. 60 is relatively new. They aren't water specific. So the more recent USP 60, it's the Burkholderia cepacia complex that you're requested to analyze. That complex could include Ralstonia piquetii as an example. The USP 62, you can see the list of organisms, principally gram-negative bacteria. So this is similar to our prior discussion. It does not mean that you have to prohibit these organisms. The monographs just give you a total plate count. They are not organism specific. You're required to understand your product, process, patient risk, and see if you need to prohibit all of these, some of these, if these pathogens could present a problem with your product. There are many other documents. I can't list all of them. Again, over 140 worldwide Pharmacopoeia, the US GMPs with requirements to clean, sanitize, maintain equipment so that you don't adulterate the product. We have some European documents listed here. And the ISPE Baseline Guide, Water and Steam Systems, third edition, recently updated. I highly recommend that for reading with a lot of supportive information for this quest we're on. Now we're going to talk about sanitization. And we're going to analyze generation systems, how the water is made. And then there is always a distribution system. So you are going to make water and send it to points of use. There's not always storage, but there is storage in a vast majority of systems. So we're going to go in more detail through the generation processes, but some are relatively low risk of microbial contamination and may not justify great expense for control, and others activated carbon, reverse osmosis. I mean, some may present a higher risk, and that is where perhaps a greater materials of construction, a better sanitization method may be justified. In storage and distribution, we have chemical sanitization, intermittent heat, hot water, continuous heat, and ozone. For purified water, chemical sanitization is often appropriate, and it always can present a little bit of risk, a low to moderate. A high risk, as an example, may be where there's no gram-negative bacteria allowed. And for water for injection, the chemical is never recommended as the primary. It could be a backup if there's a problem with heat or ozone, but it's typically not appropriate as the primary. And the other methods, which we're going to talk more about, intermittent heat, continuous, and ozone, applied right, they are low risk for purified water and for water for injection and quite appropriate. When we look at the heat, ozone, and chemical, those are the primary sanitization methods now. You will see steam is also listed, and for decades, steam was the standard, especially for water for injection, but even many purified water systems. But now there's a recommendation away from it, not that it can't work, but hot water can do it just as effectively or better in some cases for lower cost. The sanitizing agents can be applied intermittently or continuously. And in some cases, for some components where sanitization is not appropriate, replacement can accomplish that. In some cases, ion exchange resins may be sanitized off-site. Continuous sanitization, and normally this is the tank, when there's a tank, and sometimes the loop. We're talking either hot water or ozone. If the water's going to be used hot, then high temperature water is generally best. And what we mean by continuous sanitization or self-sanitizing, it's always in a lethal condition. There isn't a sanitization procedure per se, unless it's remedial, because it's always being sanitized. If the water will be generated at ambient temperature and used at ambient, ozone may be a better choice. It's a very good choice. And so there is no flush required. The ozone in water will be converted to oxygen. We'll talk more about that. And so the water is staying in a very pristine microbial condition continuously, but then is suitable to go to use. In WFI, the heat is more common, and in purified water, Heat is relatively common. Ozone is growing at a very fast pace. Intermittent, meaning that now there's not always lethal conditions to microbes, so water is typically ambient. The more the sanitization is performed, the better. When it's automated, it's more likely to be done more frequently without operator intervention. Um, it's best when you're using the water at an ambient temperature. Heat and ozone are both effective. The chemical is less effective, but it's not the point of this webinar to try to talk you out of chemical at all. If it's just purified water, if it's the 100 CFL or CFU per mil limit, chemical is proven to work effectively in thousands of systems, and it's in many manufacturing systems and many lab systems. The sanitization frequency is critical. It should be data-based, and that means looking at the product again, data, production impact, history, and in the end, you're going to need to determine the frequency that you can demonstrate that you're maintaining system microbial control. Time out of service, hot water, 3 to 6 hours generally, could be more in some cases. Ozone can be quite fast, as little as 20 minutes to a couple of hours. Chemical is generally the longest. There are a few hours contact time, but then there's often extensive rinsing time that could make it 12 hours or more. A quick view of outage time. Now let's look at what is the impact. When you pick a microbial limit, what is the impact? And you have to design to be consistently below that limit. So if the limit is 100 CFU per mil for purified water, you can't design a system that's going to consistently run at 90 CFU or 50 CFU. With microbial growth spurts, it's too easy to go out of spec. So no matter what the limit is, you're always designing conservatively. Drinking water is only 500 CFU per mil for EPA drinking, and that's really worldwide a pretty popular number. So you're not even being asked to be a log lower. So the purified water max is pretty attainable. It's not an issue for most systems, and certainly if a system is well designed, well maintained, there should not be many failures, if any failures. So this is a generous limit, a wide variety of designs, materials. It may be chem sanitization, polypropylene distribution. Dead leg avoidance is critical, and that's true really of every system that we're going to talk about, generation, distribution. Dead legs are the enemy when you want good micro control. Temperature control can be very helpful here, often overlooked. When you think of if you want to make bacteria grow rapidly, in lab cultures, it's typically done at 30 to 35 degrees C. That's to promote growth. Therefore, you don't want your system getting into a temperature range that's going to make bacteria reproduce rapidly. So now we've said 100 CFU per mil is not very difficult to meet. What if it's 100 CFU per mil purified water, but no gram-negative bacteria? And it could say no gram-negative bacteria, no Pseudomonas aeruginosa, no B. cepacia, etc. That changes everything. Now meeting the 100 CFU per mil becomes a moot point. The system is going to be designed to have no bacteria, if possible, because many of the waterborne microbes are gram-negative. So it's going to take a much tighter design. Chemical sanitization presents a significant risk for this goal. So heat or ozone become recommended. And the real issue is recovery from failure. If you have failures, the chemical sanitization only, no capability to go to heat or ozone, that's where the risk is presented. Now let's lower it to 10 CFU per mil, still in the purified water or non-compendial water domain. I think that chemical sanitization in a good design still lets you attain that, still non-metallic materials. So there's some risk here, maybe more frequent sanitization, but still you have a wide range of system choices. We drop to 1 CFU per mil, and now I would not say a simple system, a chemically sanitized system only couldn't do it. I would say there's significant risk in this because, again, if you start to fail, you may have a very difficult time getting out of it for a future consistent operation. And now heat or ozone sanitization, and it may be heat and generation. Ozone is really a distribution sanitant, not generation. But the more robust methods become recommended. 10 CFU per 100 ml, that is the water for injection requirement. Now you're really aiming for 0. That is your design. Sanitization is critical for both generation and distribution. Now when you pick high temperature water or ozone, that's going to limit your suitable materials of construction, generally increase that cost. And it's not just the process and the sanitization method. Construction details become critical, even with no gram-negative bacteria. You may well be into hygienic construction, tri-clamp joints, orbital wells, minimal dead legs again. And system maintenance, it's certainly always critical, but with really tight micro goals, it becomes even more critical. So how do we make systems that meet these microbial goals? First, you have to start with drinking water quality. In the US, it would be US EPA National Primary Drinking Water. In other parts of the world, there are other drinking water authorities with pretty equal requirements to US EPA. So you need to start with good water or make it good. If it's not consistently good, you may want to enhance the water source. That may mean adding chlorine. It may mean adding microfiltration. We'll talk a little further about that. We'll look at the process design, but it isn't just the process. You can have the right process components. You still need the right materials. You need the right sanitization method and frequency. You need the right operation. So you're going to manage external contamination, and the overall goal, again, is minimizing biofilm formation. We're going to look at the generation process units. And the control strategies we're going to look at, keeping residual chlorine where it can remain, possibly continuous circulation where there's no disinfectant. Distribution, there's always continuous circulation. There's been a belief for decades that circulation helps in minimizing microbial growth, and it may help, it can't stop it. Circulation isn't lethal. So there are successful start-stop systems, start-stop with appropriate flushes. But certainly circulation in non-chlorinated parts of the system is a consideration. Membranes are frequently employed. Of the microbial reduction processes now we're going to implement membranes, meaning reverse osmosis, ultrafiltration, submicron filters. We'll assess those. Ultraviolet light is effective. Distillation, certainly, operating above 100 C, it certainly is an effective microbial control tool. So we want to minimize unit processes that promote microbial growth. We want to maximize or put in ones that reduce the counts, and all while considering in the components periodic flushing, backwash, regeneration, and certainly sanitization. So in the United States, every municipality uses a form of chlorine, either free chlorine or chloramine. And these relatively low levels that are shown, zero point 0.5 ppm free, 1 ppm combined, often will provide a good quality feed water well within EPA. It's relatively common in systems to supplement the chlorine level if it's not consistently good from the municipality. In private supplies, it's very common to add chlorine to the well water, untreated river water, whatever that private supply is, to assure the starting water is meeting drinking water requirements and is good microbially. And we want to keep chlorine in the system, but if we look at the components, early pre-treat microfilters, multimedia, screen filters, etc., typically they're chlorine tolerant. Softeners can take maybe up to a part per million before resin life becomes bad. But the more expensive components, reverse osmosis, continuous deionization, UF maybe, but after RO or CDI, EDI, there won't be chlorine there anyway. And stills are not compatible at all. So there can be significant damage to expensive components with stills. So although chlorine helps, it can only stay in a limited part of the system. We talked about temperature control as being very overlooked in many systems. Avoid temperatures that promote growth. Below 20 degrees C is extremely effective. Even at 25 C can be quite effective. And there are cold process systems. Those are more in like the 4 to 10 degrees C range. They're cold for process, not for micro control, but the micro control is extremely effective. So cooling should be considered in generation and in storage distribution. At the start of a system, there's often a type of filtration. These microfilters, there are many different membranes and types, but they are capable of removing fecal coliform, cryptosporidium, Giardia lamblia cysts. They can give a multi-log overall microbial reduction. A common use is using where the sources are not consistently meeting drinking water quality. These are often put in to remove coliform, remove crypto, and bring it to EPA or equivalent levels. They are a very good tool. They're not an inexpensive tool. So if you can accomplish the same thing with chlorination or another alternative, you may go that way. Softeners, the most common form of scale control ahead of reverse osmosis, ahead of stills, relatively low risk, typically not a huge micro issue. The brine can in fact sanitize resin. At times, not common, but the brine can eventually become a microbial source. So actually, when you're regenerating, you may be inoculating the softeners, and then you need remedial action or adding a sanitant to the brine. So if we look at the construction, most of the time in inexpensive construction, FRP tank, etc., sanitization with peracetic acid is usually good enough. You can build softeners for hot water sanitization, no problem with the resin, but typically all stainless steel construction, and that money may be better spent elsewhere, and usually is. Dechlorination, always a difficult choice, a lot of controversy, and all of these choices have impact. So activated carbon dechlorination, it's the most common process. There will be some micro level downstream. Chlorine is gone. There can be some organic adsorption. So some micro level downstream will be likely. Hot water sanitization is very effective. And so even though there may be some counts, it's typically manageable in a total system context. And often ultraviolet light is employed downstream of carbon and is very effective in knocking those counts down if they're there. Chemical injection, typically sodium sulfite or bisulfite, it's often believed to be far better than carbon microbially, and that is possible if it is optimized, if an amount that's just above the stoichiometric requirement to destroy the chlorine is consistently there, it can be very good, but often it's over-applied, and that from promoting anaerobic bacteria growth, there can be some serious fouling problems if it's over-applied. If it's under-applied, well, now that leaves residual chlorine, which may cause oxidation issues. So the point isn't to say that it can't work. It's to say it's not always better than carbon microbially and can be less operator-friendly. And the only other real practical alternative, a high amount of ultraviolet light dosage, clearly the best microbially, because there's not only enough energy for microcontrol, there's far more to do the dechlorination, but it's the hardest to design, to determine the UV dosage, depends on the type of chlorine, the max level. And so of the three choices, when some are replaced because they haven't been working properly, I'd say the UV is the most replaced, and it's not the technology, it's the sizing is not easy. Now we're talking about UV light again, not for dechlorination, disinfection. Some people simply believe it doesn't work. I am not one of those people. I've had good experience with it for decades. It's not instant kill. It prevents replication of bacteria so they can't cause infection. It needs to be employed right. Very good after carbon, as I mentioned. It may need filtration ahead of it to keep particulate from shielding microbes from UV. But it can be very effective in a total context of a system. Reverse osmosis in the majority of pharma water systems, it's an excellent microbial barrier, an excellent endotoxin barrier. It does take the right construction. It takes the right pre-treat, the right maintenance. Sanitization is either chemical or hot water. But a done right RO is an excellent tool in this total system context. And there are non-pharma membranes. A pharma membrane that's called full fit or loose wrap allows a small amount of water between the outer wrap of the element and the pressure vessel and can help to control bacteria in that area. All hot water sanitizable RO elements are of this construction. In ion exchange, we have a few options. The top picture, continuous deionization, or generically electrodeionization. We'll talk about the microbial control attributes. Then ion exchange resin in vessels. It may be regenerated off-site. It may be regenerated in place. The picture in the lower right, the off-site regenerated can be done in stainless vessels, sanitary piping, hot water sanitized, but quite rare. So we'll compare the ion exchange options. In electrodeionization, with the electric field, there is significant water splitting, production of hydroxyl and H plus ion. And so locally, there's very low pH or high, and this is biocidal. So they do not only are they not a microbial risk, but they can actually reduce counts. And with the right construction, can be hot water sanitized. Off-site regenerated resin, it can add some counts. It can give a little bit of reduction. The hot water sanitization is rare. If the resin is regenerated in place, that's rare in the United States. In other parts of the world, it's more common. The regeneration chemicals, acid and caustic, actually can help if done frequently enough and can provide pretty good micro control. Microbially retentive filters, the most controversial component in systems. They can be part of a total micro plan. They should never be the sole solution. Regulators do not want to see a retrofit of these filters to solve a problem instead of solving the problem itself with better sanitization, better construction, better maintenance. But when you have a good system with several barriers, then these final filters, as they're sometimes called, they are useful to produce water for injection and better. They are useful to meet no gram-negative bacteria. They can be integrity tested. The 1993 FDA guide on high purity water systems does state that use should be validated, how to determine the period in use, the replacement. I will acknowledge there are some regulatory acceptance concerns, the locale. I talk with European people who say not as well accepted in European applications. The location within the system, I would never put them at every point of use. No auditor is going to want to see 50 individual filters at every use point because they're going to question the maintenance. So it's using them in the right place, minimizing them, but I would say they're a very good tool to consider. Same with ultrafiltration, the same issues, but the ultrafilters now add endotoxin reduction. And so in non-distillation water for injection systems, ultrafilters are in a very high percentage of those systems. And stills, there's not much to say. They're not a micro issue. Certainly evaporating above 100 degrees C, they provide very good micro reduction. So quickly putting some systems together, a wide range of configurations, wide range of materials, wide range of what is GMP. And the greater the risk aversion generally means a higher cost. But what is acceptable is a tremendous variation between users. Here's about the simplest system that you could use. A little bit of filtration, some carbon pretreatment for dechlorination, an ultraviolet light for microbial control, ion exchange, again in the US, typically regenerated off-site, a final filter, and continuous circulation. And there's often a heat exchanger to control temperature. This could go to a tank or it could go direct to a loop. Now, there are many variations of this. We could put 100 flow schematics up that could all work. So this is a very generic configuration. If we assess it, there shouldn't be a conductivity issue. There's typically not a TOC issue unless it's extremely high TOC feed. There is not a membrane, but the ultraviolet unit could be a TOC reduction unit. If you only need to meet 100 CFU per mil, there are thousands of systems out there like this that work. There's zero wastewater or minimal, and of all of our possibilities, they're about the lowest capital cost, and they definitely can be a contender. You could state this is the least robust system possible compared to membrane systems, distillation systems, and yet these systems done right can certainly succeed. And as I've mentioned, if instead of off-site resin, it's done in place, that enhances the micro control. Now, we could put 50 membrane systems up, but we'll take a quick look at one. Media filtration, softening for scale control, carbon for dechlorination, some filtration UV, and now starting with processes that reduce. The UV reduces microbes, the RO should, CDI, EDI should, UV. There's potentially a post-CDI UV, and it shows ultrafilter. This would be more appropriate for water for injection. There may be a final 0.2 or 0.1 micron filter. So we have several microbial control processes. This could be hot water sanitizable. It could be chemically sanitized. If we assess this with hot water sanitization, easily meets conductivity, total organic carbon, microendotoxin. There are units like this in successful operation for water for injection, for highly purified water. Excellent where no gram-negative bacteria are allowed. So this is capable of water right through water for injection, but it isn't just the process, it's the construction and the maintenance. And here we see a process like that, a skidded system, but it's all sanitary stainless tubing, orbital wells, tri-clamp joints, and hot water sanitization. So the robustness really, it's the process, it's the construction, it's the sanitization and maintenance. If this were a chemically sanitized system, it is going to still easily meet conductivity, total organic carbon, most micro limits. By the time now it's no gram-negative bacteria or water for injection, the chemical sanitization, the lack of hot water sanitization, it increases the risk substantially. But yet, there are many systems like this, especially in other markets and in pharma. But you also need to take into account regulatory expectations. So I would say in a membrane system like we've reviewed, if it is for water for injection, the hot water sanitization, I believe, is a must, both from a practical standpoint and from a regulatory expectation. In distribution, we're going to go through quickly the key elements. We'll quickly discuss the importance of velocity and turbulence, materials, surface finish, joining methods, valve types, dead legs, and sanitization. Surface finish, another great discussion. I'd say worldwide, most water for injection, certainly in the US, 15 to 20 microinches RA, and we have the metric equivalent shown after that. Purified water, more typically 25 to 32 RA. The real question is that necessary? Could it be worse? Should it be better? Many consider these to be critical. I would say consider the application. If you have your entire storage system continuously at 80 C or at 80 degrees C every day, does surface finish really matter? Do you need 15 RA, which typically you get to electro polishing? I would say no. So I would say, although these are a pretty common range, really consider the entire context. What if a tank is in continuous ozone presence? What if the loop is ozonated every day? And the final bullet really is important. No surface finish totally inhibits microbial attachment. This has been proven in many studies. Eventually, the microbes will adhere. So no matter what the polish or the surface, you still need sanitization. You still need other processes. You can't just polish your way to low bacteria water. This is simply showing in studies that have been done in microelectronics, many non-metallic polymers delay the attachment much more than stainless or even electro-polished stainless. Having said that, eventually the microbes will adhere to every one of these materials. And after that, then the growth rate is all the same. Velocity. Another thing considered to be very important, and I'm not saying that it should be ignored, but let's ask the question in your storage tank, what is the velocity? What is the Reynolds number? it's obviously incredibly slow. It could be a tenth of a foot per second, a tenth of a meter per second. So the largest volume of water in your system is not going to have turbulence at all. There's obviously going to be laminar flow. Within the loop, there's a lot of popularity to have the Reynolds number exceed 4,000. There is some logic to that. It helps in branch mixing. So even with small dead legs, velocity is going to help in branch mixing, keeping piping fully flooded. Most of the time, the velocity is going to end up somewhere in the 3 to 7 foot per second range, maybe a little lower, maybe a little faster. It can't be too high or you suffer high pressure drop. If it's too low, then you may be wasting money on pipe and fittings that are oversized without good engineering practice. Most agree that even if you're trying to have a Reynolds number above 4,000, above 8,000, if at max demand, a short period, if you're going to have non-turbulent conditions for relatively brief periods, validatable periods, that's perfectly acceptable. And you can waste a lot of time and money to try to achieve some specific velocity or Reynolds number under every possible condition, some of which may never happen. Similar to the thought on no finish is going to stop microbial attachment, no velocity totally inhibits microbial attachment. So it can be useful, but sometimes is overthought. Materials. The picture on the left is welded stainless steel pipe, typically more appropriate for sections of generation, have not seen this as a loop material in decades. The picture on the right is 316L tubing, orbital welds where possible, which is most places, tri-clamp joints where required at components, instruments, etc. Certainly in the metallic world, this tubing, the 316L, would be by far the most common. In non-metallics, which are in many, many systems, both in generation and distribution, the picture on the left is polypropylene. That happens to be IR infrared welded. So it's a butt weld joint, and they're not perfectly clean at all, nothing like an orbitally welded stainless joint. But in many successful systems, in the middle is PVDF piping, very commonly used in ozone. It's of non-metallic piping, it's the most ozone resistant at a reasonable cost. And the picture on the right, that is PVDF that's done with a bladder in. So it's heat welded, but with a bladder in place. And these joints can be smoother than an orbitally welded stainless steel joint and can make for a really smooth system. It's a great surface finish. They're great joints at a cost and could be a great mate with, say, ozonated distribution if you wanted to do a non-metallic. We can't talk about microcontrol without dead legs. Decades ago, there was a rule for a 6 diameter dead leg maximum. The diameter is the branch, and it was measured from the center line of the main run, which really made no sense and was based on a hot system, an 80-degree C system. It was based on conduction of heat. Today, when we talk about dead leg diameters, it's from the pipe wall to, say, the wear of the diaphragm. There's a common goal to be below 2 pipe diameters. And in most diameters, in most cases, it's possible. It's not always possible. In tiny diameters, fractional inch, it can be difficult. But the 2D, it's a good goal, and I can't say enough. Auditors, regulators understand unnecessary lengthy dead legs are not good, and probably merit citing, but they are a real problem, and the shorter the dead legs can be maintained, the better. Well, from a valve standpoint, zero dead leg valves, the upper picture is what is the older technology. There really is an integral, minimal T, you might say, a fraction of an inch. But the lower picture is the newer preferred, where the block is right in the piping itself, and it really is a minimal dead leg. It is your friend with chemical, heat, ozone, and really was a great thought to help us in microbial control. Good sampling, a valve that can protrude up into the pipe, easy to flush, minimal water volume is going to give you more accurate sampling. We need to now do a quick study of distribution of sanitization. So here we see chemical sanitization. This is typically ambient production, ambient use. The chiller or heat exchanger for cooling is a very good addition to be able to keep a friendly temperature for microgrowth. The ultraviolet light that's shown optional and the submicron filter that's shown as optional, I think if there's not a regulatory issue, if you can use both, they can help to reduce the frequency of chemical sanitization, still give you good micro counts. And chemical sanitization is timely, expensive, and typically not done that frequently. If we look at heat now, heat could be in via a tank jacket. It could be in through an external heater that's steam or electric. If it's continuous hot, there is, of course, no risk. Ideal if you can use all of the water hot, most of it. You may need use point heat exchangers in some cases. If we look at intermittent now, when we're in operation at ambient temperature, again, temperature control would be good. And here you have to debate use of an ultraviolet light and or a filter once again. If they can be used, they can reduce the frequency of sanitization. If sanitization were going to be daily, it's kind of a moot point, and probably those polishing components are not required. And so when it's in sanitization, chillers off, heat exchangers on, and just proven over the decades to be extremely effective. Ozone is growing rapidly. Ozone at its best is continuously applied to the storage tank. It could be as low as 20 parts per billion. I'd say the most common range is about 50 to 100 parts per billion in service. So the water in storage is always protected. Ozone cannot remain in water to production. It would be a violation of, at the minimum, the USP monograph that says no added substance, and an antimicrobial agent would be an added substance. So ultraviolet light, about 3 times the dosage for microcontrol, is implemented. There's an ozone monitor, typically pre-UV, to prove that the level of ozone is there that should be there. There's a monitor post-UV in service showing that there is no ozone there and the water's actually meeting the monograph for purified or water for injection. And often there's a third monitor at the end of the loop for use of sanitization. In the sanitization mode, which could be as short as 20 minutes, maybe a few hours, maybe an entire shift dependent on production, the ultraviolet unit is now turned off. That means that now ozone will not be destroyed in UV. It will go out to the loop. The ozone controls typically are now designed to achieve a higher level. And so sanitization levels are often from 100 parts per billion to 500 parts per billion. And ozone is proving to be extremely successful. There is an ISPE guide on ozone in pharmaceutical water. I would also recommend that for reading if you don't have it, if you're considering implementation of ozone. So now we've talked through incredibly quickly, I apologize for the pace, but we've talked about how to design systems to not get in microbial trouble. What happens if you do have failed samples? What happens if there's a possibility that you have unacceptable microbial contamination? You would want to review the test procedures for sure. You would want to view the sanitization procedures. You're probably going to conduct additional microbial tests, the number of points, the number of samples per day or week. You need to determine, can it be assessed as a sampling issue, or do you have a problem? You may well, even before those micro results are back, do an additional sanitization. You may increase the sanitization frequency. Certainly, you'd want to assess the system and see, is there a dead leg issue? Is there a source of external contamination? As an example, the vent filter on a storage tank. So it's both auditing the system to be sure there aren't design flaws, but it's really analyzing the data and the sanitization. Look, sometimes that it's not sampling, it is a problem. Maybe the sanitization temperature hold would be longer if it's heat, or a chemical hold, or an ozone level in sanitization. If you need biofilm removal, sodium hydroxide is probably the best chemical to do it. It's extremely helpful to dissolve biofilm, dissolve the EPS. Often there's then a water rinse and the peracetic acid hydrogen peroxide mixture sanitization done following that. Ozone can do it. Ozone can oxidize anything that's organic, and biofilm is almost all organic. But the tricky part is, when I say that an ozone sanitization of a distribution loop could be 20 minutes, that's because in a well-designed system with daily sanitization, there's almost nothing there to work on. If you have direct contact with a bare microbe, it's probably destroyed in seconds with ozone, studies would show. But if you have mature biofilm, it's not going to be seconds, it's not going to be minutes, it may not be hours. So it may be days if a system, and this would never happen in a system that has ozone built in. Ozone is also used, like sodium hydroxide, in remedial work of systems that do not have continuous ozone. So the ozone can be far faster than chemical for intermittent work. The materials need to be compatible to do that. It can be used in remedial work if it's not present, but it may take a significant time for a bad biofilm. The sodium hydroxide may be a better choice. So we have covered a lot of material. I'm going to turn it back to Rich. He can explain how you can review the material again. So Rich, I turn it over to you.
Thank you, Gary. Very informative session. I hope that the people listening in also took away as much as I did. Sorry, we don't really have a chance to answer questions directly here, but we will respond back to any of the questions that we have received. And if anyone would like more information, feel free to reach out to me at richard.jarrett@evoqua.com. We'll also have a recorded version of this webinar on the Evoqua website that you can go back and listen to. And I'd also encourage people to, if they're interested, listen to our previous webinars on sanitization and membrane water for injection. So I'd like to thank everyone for your time. And once again, feel free to contact me directly if you'd like further information or any assistance from Evoqua on your water needs. Thank you.